Hello, my name is Gary Kunkel from the Department of Biochemistry and Biophysics at Texas A&M University in College Station, Texas. Broadly, my lab studies the process of transcription in eukaryotes. Transcription is the first step in gene expression, and therefore is the point at which much control occurs in order to differentiate which genes are transcribed in an organism. As the diagram shows, genes contain promoters and enhancers. These are DNA elements that are landing sites for proteins that regulate where transcription occurs, in what types of cells transcription can occur, and how efficiently it proceeds. In all, you can see by just some of the blobs, which represent proteins on this diagram, this is a very complicated process involving many, many different proteins. Furthermore, not shown in a diagram is the role of chromatin proteins, because the genome is associated with many proteins called to make chromatin structure in the cell. We focus on a particular transcriptional activator protein that goes by several names. One name is SBF, another Staf, a third name ZNF143. These all mean the same thing, just different names for the same protein. SBF Staf is a very interesting transcriptional activator, because this protein, as shown in the diagram, can transactivate distinct kinds of promoters using two different so-called activation domains. One type of activation domain will transactivate small RNA gene promoters. A second will transactivate promoters which encode messenger RNAs. Ultimately, we would like to understand and compare the ways in which SBF Staf works on these distinct promoter types. It's very important to investigate transcription in a whole organism. Therefore, we have elected to use zebrafish as our model system. Zebrafish is a model vertebrate organism. SBF Staf is only found in vertebrates and not in other metazoans such as Drosophila and not in yeast. Therefore, it's a very good organism for our studies. The diagram shows how gene expression can be manipulated in zebrafish embryos by the injection of antisense oligonucleotides to knock down expression of very specific genes. We inject such reagents called morpholino oligonucleotides, or MOs, to reduce the amount of SBF Staf protein in the developing embryo. As the diagram shows, this knockdown results in a range of mutant phenotypes. Therefore, SBF Staf is important to control development, presumably via transcription of certain target genes. We've discovered a few important target genes or promoters that are controlled by SBF Staf in zebrafish embryos. We would like to expand this list, but most importantly, we would like to understand how SBF Staf, or the mechanism of SBF Staf, at distinct promoters. What other proteins does it interact with, and how does it actually work to control transcription in the cell?